Welcome to the second part of the lecture. In this part, we will look into electron tomography. The intended learning objective of this part of the lecture is to gain an overview about single particle reconstruction, familiarize the basic principles of TEM tomography, application of electron tomography. I will give some of our own examples, sample preparation, processing, and analysis, some of the limitations of TEM tomography. Let's have a look into these two images shown here. This is a shadow where a man is walking downwards, and in the right side, what we see a small child with a spoon in his left hand. But we have certain limitation to understand what exactly these two shadows are. The reason is that we have information only in 2 dimensions. We miss the information from the other dimension. This gives us a limited information. In reality, these two are from exactly same object, depending on from which direction there is a light and from which direction you are viewing. That means to understand and gain information of any object, we need 3-dimensional image or details. There are numerous techniques and tools to image in 3 dimensions, from molecules up to entire or whole organism. In this context, electron microscopy actually is unique because it covers from molecules up to a cellular level. If you go at higher length scale, there are numerous other techniques routinely used in medical technology. And very often, as we move towards higher length scale, we have to compromise, of course, with the resolution. At the same time, we can use more than one techniques or combination of multiple techniques to study at different length scale. Let's start an example what happened in Natal region of Brazil in 2014, where people developed strange symptoms with a flat pinkish rash, bloodshot eyes, fever, joint pain, and headaches. And with the time, this has spread around other regions of Brazil and South American countries. And it is because of the Zika virus, which in 2015 May predicted to have more than 4 million cases. This resulted in children born with the deformed head. 2 years later, the first structure of this Zika virus was reported, and this structure was purely based on cryogenic TEM on Cryo-TEM single particle reconstruction method. A year later, high resolution with more detail was reported. So understanding this structure purely by electron microscopy imaging means that microscopy has moved from simple imaging and analysis tool to a complete structure determination tool. This is a tremendous revolution that happened over the last 1 or 2 decades. Let us look at the current crisis and the pandemic. Early January 2020, it became public that about coronavirus, and it of course based on numerous other techniques, and one of the techniques is also electron microscopy imaging that confirm the coronavirus structures. 2 months later, a bioRxiv paper appeared showing the first 3-dimensional structure of SARS-CoV-2 spike protein, which is fundamentally important, for example, developing vaccine. Less than a month later, this paper was published in Science. Now, if you consider Zika case, it took 2 to 3 years to report the structure. And in coronavirus case, since it became public, it took only 2 to 3 months to publish the data, which essentially means that around 6 years' time, the progress in imaging, image processing, and computation has grown so rapidly that today, a structure determination of biological particle has become very routine. This has to do to the development that happened in 1980s, especially from Joachim Frank, who also received the Nobel Prize in Chemistry in 2017 for Cryo-TEM development. He developed this single particle reconstruction, where you acquire images, a large number of images, and collect 200,000 to 1 million of particles. Ideally, it applies if your particles are absolutely monodispersed, such as virus particles and proteins. They're atomically precise. When you have a large number of particles, as we have seen in the previous lecture, that Cryo-TEM sample preparation, you can rapidly freeze, and then particles can be imaged with the different orientation. Then you can collect all those have same orientation and classify them into one group, and then you average them and determine the structure. Here is a video from Gabriel Lander from Scripps Research Institute, where shown, let us imagine that we have an aqueous sample. Again, I'm showing the same Cryo-TEM sample preparation, where particles or nanoparticles are randomly moving. In this case, it's a Groel complex, which is a chaperone that actually controls, for example, protein folding. Now we need, as I said, only 3 microliter of sample. This sample can be then plunge freezed using WIT-robot, and then we generate extremely thin ice. Once you generate these thin ice, you can then image them. Now, under room temperature, particles are randomly moving, but when you plunge freeze them, now you have vitrified ice and different orientation of particles are frozen. And once you image under the microscope, what you will see is that if you have now 3 different orientation of these particles, when you image them, you start to see slightly different projections. That means they have random orientation, they produce different projections, which means if you have this kind of TM image, now you can categorize them. You can see that, for example, these particles, they have similar orientation, whereas there are other particles like here, they have a different orientation. So you can classify them and group them together. Once you group them together, then you can identify and sum them and combine them and create the final structure. And this structure not only gives you a isosurface image, but if you know the protein sequence, you can also dot the structure and you can get atomic resolution of few angstrom. It means that you could be able to see an aromatic ring in these cases. So this way you can solve the complete structure of particles. It involves sample preparation, specimen preparation, acquiring raw data, processing the data, classifying and image processing, and then 3-dimensional structure determination. The advantage here is that you need a very little sample. There is no need to crystallize, and the entire process happens. You can directly extract the biological sample or even synthetic nanoparticle or embedded nanoparticle if they are atomically precise, and you need only few microliters. For crystallization, you need a large number of samples, and importantly, many of the particles do not crystallize. Let us move. What if we go even higher structures? Single particle reconstruction applies only for biological particles because they are atomically precise, they have better symmetric structures, and they have sometimes often repeat units which are same. Therefore, it is easier. As we move to synthetic materials, metal nanoparticles, and other assembled structure, we start to go for more polydispersed, different size and shape, and how to study them, how to study those non-uniform structures. Let us consider a sphere, a disk, and a rod. Now, if they are oriented in this way, because transmission electron microscopy, we see the 2-dimensional projection of a 3-dimensional object, we start to see all of them show the same 2D image. So conventional TM micrographs represent 2D projections of 3-dimensional object. Now, if I rotate the sphere, disk, and rod, only disk and the rod will show a different orientation. And sphere will not change, and it will be exactly the same kind of 2D projection. We start to see the disk and the rod. So what does it mean? It means that this 2D projection in TEM depends on the orientation of the object. So if we can summarize, we have two-dimensional projection, which depends on the orientation of the object. 2D projection contains also tremendous amount of internal structural detail because it's a transmission mode. Unfortunately, due to overlapping information, only limited structural details can be extracted using simple conventional 2D projections. As an example here, how the orientation dependency affect, this is a origami negatively stained structure. You can see this rectangular structure everywhere, but then there are two outliers which shows a different orientation. They're exactly the same structure, but they're oriented differently. If you have some higher order spherical structure, we can see that this one has low contrast at the center, that is the core, and then we have higher contrast in the shell. There is a uniform core and the shell, and some random pattern that you can see in this. So for that, what we can do is that electron tomography. We are all familiar with computer tomography or MRI imaging. What happens in computer tomography is you have the sample or the patient here, and then we have source and the detector, where the source and the detector are rotating, where the sample is stagnant. In this way, you can collect the slices or images at different angle, the entire 360-degree. So this helps you to collect a series of these slices, which can be stitched together, and then you can build a three-dimensional structure. Same is also in MRI. But unfortunately, we cannot do the same in TEM because of several reasons. We cannot rotate the source and detector. We can only rotate our sample or TEM grade. And in this way also brings a lot of limitations. Here, source and detector are stagnant, only the sample is rotating. Because the TEM grid will block the beam above certain angle, we have limitations also to rotate the sample. We can only rotate to a certain degree, such as plus 70 degree to minus 70 degree. Of course, there are these days, it is possible to have continuous rotation, even though it doesn't help much. So the 3D reconstruction of particles was first formulated already more than 50 years ago by Rosier and Klug. They have formulated principles of 3D reconstruction of using a set of EM images. So what is tomography in general? A simple definition for a tomography is the construction of higher dimensional structure using a series of lower dimensional projections. So if I have a series of one-dimensional projection, then you construct a two-dimensional structure. If you have a series of two-dimensional projection, then your reconstructed or tomographic structure is three-dimensional. In TEM, by default, we collect 2D projection. Therefore, TEM tomography means 3D reconstruction, or these two are essentially the same. How we do? We have object of interest here on the grid. It's a three-dimensional object. Then you tilt from minus 69 to plus 69, and you collect image at a certain increment angle. The increment angle can be 1 degree, 2 degree, or even less or more. Generally, it is good to collect as many images as possible. This entire series of image is known as tilt series. The original 2D projection of this particular particle is here. And now, once you acquire this image, you can do some processing and it's called that weighted back projection, and which then further processing, you can construct the three-dimensional structure. Therefore, it is a 3D reconstructed 3D structure. It involves tilt series collection, alignment, 3D reconstruction. There are several important precautions to be taken when you collect this. The first one is sample preparation. When you do sample preparation, you have to identify the correct grid, the mesh size, and support film. Specimen preparation should be in such a way, if it is conventional imaging, then there should be a minimum drying artifact. It's important to know that where is the location of your object of interest that you are willing to do 3D structure determination, ideally at the center of the grid. The orientation of the object of interest and the specimen thickness and data acquisition, that means a large number of projections are the better, and alignment and the good alignment of the raw data, and then methods that you use to reconstruct. So grid selection, it's important to have lower mesh size. That means it gives you a larger area and more option to image particles. Or you can use hexagonal grid that again helps you, unlike square grid. It will not disturb as much as a square grid does in blocking the electron beam. And specimen preparation, generally the grids are preloaded with some fiducial markers. These markers are important to align the images, such as here is our object of interest. We have a lot of small particles. This is a gold nanoparticle. This acts as a fiducial marker so that we can use this to stitch the objects together. The object of interest should be clearly visible without any overlapping object. If you have another object that's overlapping this, it will not give you good data. It will be challenging to acquire the image. Location and orientation of the object. If the object is isotropic, such as a sphere, it generally doesn't matter whether it's along the tilting axis or not. If we have an anisotropic object, something like this, if this is the tilting axis, then it is good, it is oriented this way, so that when you tilt around this angle, it undergoes minimum deformation. So we will have less artifact. So the tomography involves collecting a series of 2D projection, alignment, 3D reconstruction. In between, it also has data acquisition, data processing, reconstruction, and image processing. It utilizes many softwares such as automated tilt series collection or manual collection, then processing this data, and then 3D image processing. So essentially, imaging is a small part, but processing is also as a larger part in 3D reconstruction. How it looks like here is an example of our own example of a gold nanoparticle which is peptide capped, which is nanoparticle which arranged into a spherical structure. And the image here shown is combination of about 70 images stitched together. And then once you reconstruct, you can artificially color them. In this case, the color, which is red, means that it's high contrast, generally metals. Then you move down to yellow, green, blue, and white with a lower contrast. In this case, you can see that high contrast area is a gold core. It's a cross-sectional view showing a one-layered shell. And then we have less contrast organic material. And this sphere is encapsulating some of the unreacted organic material inside this. So we can use to determine the structure arrangements of the atoms. We can go beyond distinguish if you have a heterogeneous population in the same synthesis of nanoparticle. This is a cobalt nanoparticle synthesis. It forms nanoparticle, assembles into higher structure. If you take low magnification, larger image, you see a lot of spherical particle, different size. But the difference here, you see that one which has less core, less contrast core, the others have uniform core and cell. So it is interesting to see why we see this type of structure. So if you take the TM electron tomography of this, you start to see that it is a hollow particle where the interior is empty. That explains why this core is there. That's not all. We can also precisely determine the thickness of this cell, and it's 3 to 4 layers of nanoparticle. If we go a little bit higher, we can take a small portion of the cell, and from the cell, we can determine how each nanoparticles are arranged, as we can see individual nanoparticle. It also shows the amorphous structure, which are organic material. You can take them out, and individual nanoparticles are not uniform, as you can see the size variation. But we can also do beyond this. We can pick similarly sized particle together, which is an averaging, and then we can determine the size and shape of individual nanoparticles. So electron tomography can give a 3D structure, internal structures, and the packing pattern. Now we know the location and orientation in 3 dimensions. This we can collect the coordinates from these 3D coordinates and then use that for reverse engineering or even do computational simulation. The other interesting is that if you have a mixture of more than one type of nanoparticle, make superlattices. Superlattices are periodic structures of layers of 2 or more materials. Typically, thickness of one layer is several nanometers. So here is an example. We have a virus, rod-like virus nanoparticle, tobacco mosaic virus, which is 300 nanometer in length and 18 nanometer in diameter. It has negative surface charge, and this is full of coat proteins. Now, if you take a cationic synthetic gold nanoparticle, which has surface charge positive around, and there's narrow size distributed, if you mix them, you get a superlattice. And here is how, if you mix a different ratio of gold and virus, you end up getting different colors, which indicates that there is an optimum ratio where you can precipitate out the colloidal dispersion. First, typically it's done is how the structure, to gain some structure factor, you do small angle X-ray scattering. Small angle X-ray scattering can often cannot reveal if there is a new type of structures. Of course, there is an optimum ratio. It suggests that at this stage, we start to see certain peaks, diffraction peaks, that suggest that, okay, the structures start to form. If you do the TEM of these optimum ratio structures, you start to see that higher order structure are nicely arranged superlattices, and we see some free virus particles. And if you do higher order structure, you start to see stripes. Essentially, these are virus and nanoparticles which are arranged together. Now, if you do Cryo-TEM image at a different concentration, we start to see something beautiful. We have virus particle here, and then you have gold nanoparticle. They are intercalated. As you increase the concentration of gold nanoparticle, you start to see they're tightly packed, and then these start to grow into larger structure like this. So essentially, Cryo-TEM, which is time resolved TEM, gives us a lot of information about the mechanism, how these two types of particles interact and grow together. And in the first case, we don't get random structure. It always, the gold nanoparticle goes and binds and nucleates and intercalates and brings together two virus rods, and then it aligns the two particles. Then the next particle comes and attaches very close to where you have a nucleation center. And this process is called a zipper mechanism. It works just like a zip. The nanoparticle here works in the same mechanism. But small angle scattering often gives you a lot of information, but if there's a new structure, it's challenging to explain why we observe the correct structure. If you take rod-like particle, it is expected that they form hexagonally close-packed structures. But in our case, what was observed that we have a square lattice. And that is rather surprising how the rod-like virus particles can form square lattice. For that, what we look is Cryo-TEM and electron tomography. It is the same virus nanoparticle hybrid long chain, just millions of nanoparticles arranged together with large number of virus into higher order chain. What you see here is also some helical structure that is formed. Then if you do three-dimensional reconstruction, in this case, because gold and virus have different contrast, we start to see only gold nanoparticle here. Of course, by adjusting the contrast, it's possible to look at the virus particle. But from this, we have individual gold nanoparticle. We can extract the location, orientation, and position of the nanoparticle in three-dimensional coordinate, which can then construct the structure. Now you can see why this forms square lattice. This shows from the tomography, which are colored into red, or gold nanoparticle, how they are oriented, and the space between them fits exactly to the diameter of virus nanoparticle. We can use them to study further and also understand why there's a twist, and use this coordinate also to do simulation to understand the entire growth mechanism. So here what we see is that electron tomography, not just to see the internal structure, not only to show how they are arranged, but also to reverse engineer and understand the effect of size and shape and the growth mechanism of nanoparticle hybrids. Then we can do a little bit more. We can change now. We know that if we use certain size, we get certain type of assemblies. We can change and control the size of the nanoparticle. Since the virus particle size is constant, we can change different types of nanoparticle. Of course, by tuning this, we can go and change from square lattice to hexagonal lattice. So this is the amount of information that we can get from TM imaging, especially from electron tomography. Electron tomography has a limitation, as I said, at times, samples should be thin enough, high-thick samples, of course, it's a challenging, should be mechanically robust for handling. Because we are talking about 60 to 70 images, that means it has undergone large amount of beam damage. To prevent that, we use a low dose imaging. Why the sample should be as thin as possible is because if you have a sample which is as a zero tilt, it has exactly the same thickness. If you start to tilt the sample about 45 degrees, then the thickness increases by square root of 2. And then if you increase a little bit further, if you look at here, only 15-degree change. As we move higher angle, then increases by 2 times. If you go to 70 degree, then it increases 2.9, 3 times, 80, it is almost to 6 times. That means if I have a 100 nanometer sample at 0 degree, and if I go to tilt 60, then it is eventually has to go through 200 nanometer thickness. This actually is problematic to gain good image, as the focusing becomes very challenging. So unfortunately, not all the samples fulfill above criteria. So often you can also ask, what is the resolution? Single particle, we can go atomic because it's a clear structure determination. We are talking about larger structures. And the resolution can be defined based on the diameter of the object, the number of projections. For example, if you have a simple spherical object of 200 nanometer in diameter and you have 70 projection, the theoretical resolution is 8.9 nanometer. However, let us look into this TM image. It is a 2D projection at 0 degree angle, and you see absolutely nothing here. If I tilt from to plus 68, and collect series of image, go to -68, then you start to see that it's a spherical object. And this area here is the one which is attached to the grid. So this is slightly an artifact during the sample preparation. But if you do electron tomography, you start to see some of the beautiful structures. And even we can see a few nanometer size. This is a protein assembly. And we can even see the small protein assemblies here, which form onion-like stripes. So sometimes tomograms can beat the formula. There's always formula, but often they can beat the formula. It's also possible to go beyond a simple resolution, even to monitor a single atom dislocation in 3 dimensions. And this is called atomic resolution electron tomography. And in this case, we can identify individual atoms and each atoms, and then you can construct the 3-dimensional structure. You can computationally then simulate and understand any defect. This is very important, those who study catalysis, especially heterogeneous catalysis or metal-based catalysis. There are numerous advanced methods. What we look into is the electron tomography and single particle reconstruction. There are more crystal-based called 2D electron crystallography or microcrystal electron diffraction, micro-ED. If you're interested in this topic, I suggest you read this beautiful article about electron diffraction and various methods that's used to study structures. In summary, what I can say that 2D TEM projections contain a lot of internal details of an object under investigation, but we can only extract limited information from 2D projection. Tomography offers a self-assembly and mechanistic pathway of colloidal superlattices, morphology and internal structure. It provides a lot of useful data for computation simulation and complementary tool for SACs and other analysis.